Welcome to Biology and Bioengineering Summer Camp, BBSC 2021.
All right, very good afternoon, everyone. I hope you're all getting ready for the biology and bioengineering summer camp, which is going to happen next week. But as you have already seen that, we really wanted to make an effort to make most of it more serious, to get really prepared for doing some experiments. And in that light, we are conducting several pre-workshop sessions. Aim is here to make all of you comfortable, make you more competent to do some computational analysis and some bioinformatics based work which you can do from your home. And there's a lot to be done actually, rather than thinking about that biological experiments can only be done in the wet lab. I think what we are going to convey you. I request everyone to please mute yourself. It's getting slightly disturbing.
This.
All right, so I'm saying that there is a lot can be done nowadays with the various online tools available, different softwares, and so much data which is publicly available, which you can actually utilize and do a lot of interesting analysis and project yourself, right? So in next 3 days, today, tomorrow, and after tomorrow, we are going to conduct 3 tutorials, which is essentially what also we are going to conduct in the main summer camp. But then we would expect you to already have the background and the basic prerequisite, which you are going to cover in the next 3 days. So during the actual summer camp, you will be well prepared to take the knowledge. Otherwise, just conducting a session for an hour is very superficial in my mind. You can't make somebody comfortable doing something at on their own with the confidence. It cannot be just achieved in that one session. So today we are going to have the first tutorial session. You have probably received our emails. There are 4 different aspects which we would like to cover today. Essentially, we have very heterogeneous and very broad audience nowadays for this particular session. We have students from class 8th, 9th up to different levels of BSc and MSc. So some things might be repetitive or something might be too basic for the advanced students. But don't worry for the advanced or the senior students. Even for you, we are going to raise some difficulty level in the quiz. So while today at the end of this session, we are going to have the Google form based quiz for all the 3 groups, which will be junior group, middle group, and the senior group. For senior group, we are going to give more like what you can do and then answer questions from that. So even if you know a lot about cloning and bioinformatics and primers, I think we will test your knowledge. So please be ready. But in terms of the basics of it, some of these things might be appearing some basic concept to you, and I please bear with us because we want to take the really young crowd here from the 9th to 12th to make them comfortable at the early age to know some of these bioinformatics concept and analysis. So different type of thing which people do in the basic bioinformatics, especially looking at where is the SARS-CoV-2 gene sequence, what kind of proteins it has like spike protein, and what could be sequence of that, where the mutation is happening, what could be sequence which is changing with the mutation. A lot of these things which you are hearing, you have no idea that how people can do all that, right? And which is actually very simple way you can do these things yourself using online tools. So first module will be based on this particular basic bioinformatics. Then some concept of gene cloning, how to make multiple copies of the genes, and also how to utilize the polymerase chain reaction. Different reagents are required, especially the primers. How to design the primer sequences are very important, and that you can actually see apparent in terms of the SARS-CoV-2 detection. You really want to detect only the pathogen of interest. So the gene has to be really unique or specific, which you want to amplify, and that you can only achieve using the primers. So that's the primer designing and primer sequence becomes important. So some of these things are going to be covered shortly. Let me invite from my group Dr. Arup Acharji. He is a senior research associate scientist in the lab. who's going to brief you about these four modules and what contents have been covered and what is expected of you to know about it. And then from my team, we have Akanksha and probably Dr. Manisha, who will be taking you to some of the very basic tools of how to do these analysis yourself. What we really request you to be ready with your laptop or computer, so that when we ask you to do something, you should also be able to do some of these analysis. We are also going to paste a link here in the Google Drive link,
Okay.
where in that Google Drive link, some files are there. Please download those files and be ready. All right. So good luck, enjoy the session, and we will really would like to see that you are doing the work, because all the efforts are being made just so that you can start doing something on your own. I'm sure there are a lot of lectures already available online. We are also uploading the tutorials and videos online. But what we really want to break the ice to really do something with you, right? So please start doing these things on your computer or a laptop or iPad, whatever you're using. And in the actual summer camp session, we are going to give you some assignment and test, because we would expect you to have gone through these things with the same level of seriousness what we are making effort. All right. Requesting all the participants to please keep yourself mute. And let's now invite Dr. Arup to take the session further.
Can you hear me?
Yes, please go ahead.
Yeah. My screen is visible, I guess?
Yes, please go ahead. I will stop sharing. Yeah, go ahead.
So hello, everyone. I'm Arup, and today I will be giving a brief overview of what we will be covering in tutorial 1. As Professor Sanjeeva already mentioned, that what we would be studying briefly. So we are here to give you an idea so that you can later on go out and explore. And again, you can, if you are having problems, you can come back to us, because these modules itself are so big that you can have a master's degree in any of the 4 modules. But we will try to be brief and give you an idea how things work. So first part will be bioinformatic analysis, then second would be gene cloning, third would be PCR, and then a variant of PCR that is we are using nowadays a lot, that is RT-PCR. So the first question that you would be, you would be wondering, that why is bioinformatic analysis important? Now the question is, this word itself has 2 components. One is bio, another is informatics. So the data that we are generating for the experiments that we are doing are too many. Like the data that we are generating, especially from genomics and proteomics, there is so much of data that it would be humanly impossible to actually understand all of it. So we are applying few computational tools to understand and make sense of that data. So what biological sense does this mean? So what does bioinformatic analysis help us? First of all, it tries to, if you do if you would slowly, I mean, you would be explained the tools such as BLAST and multiple sequence alignment. So with these tools, what it can help you to do is that it can help you understand how a particular gene is related to another gene. So suppose you have hemoglobin. So we have hemoglobin, even dogs have hemoglobin, fish has a hemoglobin variant. So how do these proteins, are these proteins related to each other? How did these proteins evolve? So, understanding the relation of these proteins, that can be, we can understand those relationships using multiple sequence alignment and, understanding clades. So I don't know why these annotations are coming up. Vishwa, can you just disable annotations?
You have to actually go and disable annotations. If you go to the top.
Right. I have.
Good.
So next is analysis and prediction of gene and protein structure function. So suppose you are given a new gene in the sense you have discovered a new animal. Just in the case of COVID, we had discovered a new pathogen. So how do we know that how this pathogen works? Now, much of our understanding of COVID-19 and the SARS-CoV-2 virus, I'm taking that example because it is something that is happening right in front of our eyes. So much of our understanding of SARS-CoV-2 spike protein, how it binds with ACE2 receptors, all these were predicted using computational tools. The genome that we got, the entire genome of COVID, this was, we got the first genome, I think 2 or 3 weeks into the pandemic. And how did the scientists do that? With the help of bioinformatics tools. So bioinformatics today is one of the most powerful tools that you can apply to understand and predict a gene function. On it can also start predicting how the protein structure will look like. How will the, and since once you predict the structure of a protein, you will also start predicting how the protein will look like. I mean, how will it function? So imagine you have identified a small microbe. All you can do is apply a sequencing technology, sequence its entire genome, and from the genome data, you go back to the database and start searching. Did I find anybody like this earlier? Did other scientists find any organism like this earlier? And if nobody has found it, then you can start using these bioinformatics tool to do, understand how this microbial organism is related to others. So right from discovering microbes, and also in the future, if we need to, if we go out and explore the space, if we find some organism out there in space, even then bioinformatics will be of extreme help to us. And next, another of the few functions are like prediction and detection of gene regulation networks. So we know that, suppose the cell on your skin has the same genome as the cell in your eye, as the cell in your kidney or your liver, but the cell of your liver is different than the cell on your skin. So why do these different cells behave differently, although they are having the same genetic information? So this kind of, regulatory networks that alters gene regulation. Hello, am I audible?
Arupia, please go ahead. I was unmuting everybody.
Right. So so these kind of stuff can be done using bioinformatics. So the next slide is gene cloning. So why do we clone a gene? To get more copies of a gene. Again, the question would be, why do we need more copies of a gene? Because we need to manipulate a gene. What you will understand in gene cloning is a part of cloning involves making multiple copies of the same gene. Now, once we have multiple copies of a gene, then we can start, doing experimentations, make a recombinant DNA. And why do we need to make recombinant DNA? Might be you are having a crop that is growing in a drought. So you are having a particular variety of rice that grows very well with little water. And you have another variety of rice that grows very well even if it is infested with a particular pest. Now you want to make a rice variety that will be growing very easily in less water, and also if it is affected by pest, then also it can prevent itself from this infestation. So how do we go about it? Normally there is no way of transferring one gene of this rice plant to another, like make a copy of these two genes and mix and match. So but if you use gene cloning technology, what you can do is you can clone the gene responsible for giving drought resistant to a rice plant. You can also make a copy of the gene that is giving pest resistance to a particular rice plant. And then you can make a recombinant DNA. So in one DNA, you would have a portion that is giving it resistance to drought and another portion that is giving it resistance to pest. So these kind of technologies has been used widely. It makes, I mean, people, you must be familiar with the term called genetically modified organisms, GMO crops. People say that there's a lot of, ethical issues regarding GMO crops. But what about other applications? Applications like insulin. So many of our family members, suffer from diabetes, and they need to purchase insulin. Now, earlier, when people had to go for insulin, they didn't have insulin because insulin was basically taken from other animals. So it would have, slaughterhouses from where insulin would be purified from beef and horses. So basically, what you took was a homologous protein from another animal and try to save yourself. But an advent of cloning technologies and recombinant DNA has allowed us to make human insulin in bacterial cells. So this is the power of cloning technology. So once you have the genome information that, okay, this part of the human DNA codes for insulin, you can take that region from the human genome, paste it in a bacterial genome, and now the bacteria will make the human protein for you. You don't have to kill any animal, you don't have to slaughter any animal and then extract insulin from their pancreas. So these are the advances that have helped propelled, better healthcare facilities for many. So many of the drugs that we are using that are enzyme based are being grown in bacterial cells, and it is not necessarily been synthesized from an animal source. Next is PCR. So why do we need PCR? So just like we were talking of copying, making copies of the genome, I mean gene and cloning. So PCR, you can say, is in vitro cloning. So what helps is, first of all, PCR is a very rapid technology. For cloning, you have to use restriction enzymes, cut and paste DNA, then allow it to grow inside a bacteria. It almost like a week long process. But with a technology such as PCR, which is basically doing what cloning does, but it does it very rapidly. How does it do it? So here you do amplify the DNA by using specific set of primers and buffers and salts and enzymes. So you use a thermal cycler, an instrument that can rapidly control its temperature. So you have a temperature at where the denaturation of the DNA will happen. Then there will be a annealing temperature set where the primers will come. So why do we need primers? Because the DNA polymerase cannot start. So we need to define the boundaries of which particular gene we need to manipulate. So we can fish out a particular genome from an entire range of genes found in a genome. Using PCR, we can rapidly fish out only that part of the genome which we need for ourselves. Suppose we want to make a variant of insulin. So I'm keeping this example constant and insulin. Suppose you want to make insulin. So you take the human genome, design primers for the insulin protein, for the insulin genetic sequence, and immediately you can get your own sequence of insulin. Now you can take that insulin and put it in a plasmid and make a that means involves making a recombinant DNA, and then you can put it in an expression vector, but it will make a particular protein for you. So then how does RT-PCR change the game? Now, if you understand a bit of genomic structure, our entire genome is not expressed. So there is areas of the genome that are expressed that are called exons, and there are parts of the genome that are not expressed. So those regions are called introns. So when scientists started cloning, they found major difficulty was when they were trying to clone a gene. They would clone a gene, but there will be regions of the genome that they have cloned, but they have no basic function, as so to speak. How does RT-PCR change the game? So you would know that under normal central dogma of molecular biology, people tell that we have genetic material as DNA. From DNA, we get RNA, and from RNA, we get protein. But RT-PCR involves the use of an enzyme called reverse transcriptase. So these are primarily present in viruses. So what we did was we took a viral gene, copied it into a bacteria, and made lots of that viral protein that is called reverse transcriptase. So there are multiple reverse transcriptase that are today found among multiple pharma companies and biotechnology companies, they manufacture multiple kinds of reverse transcriptase, but almost all are taken from viruses that originally had the reverse transcriptase enzyme. So once you have the reverse transcriptase enzyme, you can make DNA from RNA. And the processed RNA does not have any exon and intron. So it's all exon, exon, exon. So you get the completely spliced out variant of a gene, which does not have any intronic sequence. It's completely exon. So this exonic sequence you can rapidly clone, rapidly amplify, clone it into a bacteria, and then amplify your gene of interest or make your protein of interest in an expression vector. Also have detection of pathogens like bacteria, viruses. The present COVID-19 pandemic has been caused by a virus called SARS-CoV-2. Now, these viruses are basically having RNA as the nucleic acid material. Now, if you want to detect this virus, traditional PCR won't be able to detect it simply because there is no viral DNA, it's viral RNA. So how do you detect it? Here you take the virus, extract the nucleic acid, apply a reverse transcription step, and once you are done with the reverse transcription step, you are having complementary DNA. That DNA can be again amplified using traditional PCR. Then that will help you to detect pathogens. And you are seeing on the televisions that every day we are getting around 3.5 lakh to 4 lakh infections. So most of them are detected by using these very technologies. So these technologies, just think of it, if we would have had a COVID pandemic about 30, 40 years ago, there was no way we could have detected the virus until it would have, wreaked havoc into our body and gave us symptoms, and we would have become, we would have antibody tests, of course, but even then, like, it would have been much more difficult. We wouldn't know the sequence of the virus. We won't be able to design a vaccine against the virus. So a lot of these technologies are, these bioengineering technologies are helping us right now as we speak, and it has affected our life greatly in the beneficial sense of the term. So with this, I will will end my talk. So there are tutorials and hands-on that are coming up where you can, get a real sense of how to work on these methodologies, how to use this for your own benefit. Thank you.
All right. Thank you, Dr. Aru, for giving a very good overview. I think the participants can actually, ask questions in the chat box if they want to ask something. Ankit, please enable the chat. And let me now request Akanksha to start the hands-on session. Akanksha, please share your screen and give instructions to the participants.
Yeah, thank you. First of all, okay, so we'll be covering very basics about bioinformatics, and we'll start with retrieving the sequences. So let me share my screen. Okay, I am not able to share my screen, so I think I should be given permission to do that.
All right, we are doing that. Please go ahead.
Yeah. Okay. Okay, so first you go open your search engine, and you can either type NCBI, or if you have the link, which is ncbi.nlm.nih.gov, you can directly go over there. But when you Google search for NCBI, this is what you get. NCBI stands for National Center for Biotechnology Information. This is the site that maintains the database GenBank, and we'll be retrieving nucleotide sequences. So this is how the web page of NCBI looks, and this has been covered in the video as well.
Akanksha, you have to disable annotation. I think we are still in a group of children who are really, many of them are still very young. So can you figure it out?
Yeah, yes, I'm figuring it out. Just
on the top, if you go in the screen share part, you can go disable annotation. By the way, participants who are disturbing this kind of activity, we are going to remove you from the session.
I got it. Okay.
So tracking team, please make sure we disable those participants and remove them from the workshop.
So now from the NCBI database, we can either take, so we can search the whole database for the particular protein or RNA that you want. So either you can directly go for protein, be specific and go for the protein or the nucleotide sequence, or you can just type, let's say if I talk about spike protein.
It will be good for the participants to do these activities on their own computer so that they can get benefited.
So what I will get is all the information that NCBI has for coronavirus spike protein. So either it will ask me for the gene, or if I go down, I will also get the number of literature that has been published for it, the gene sequences, the genomes, the clinical trials that have been going on, and various databases where they have been submitted, and other structures and compounds that are related to it. Okay, so now if I want to go to the nucleotide, I will go here, click on the nucleotide and go for the nucleotide sequence. Okay, so now I'll be getting all the coronavirus sequences from different, it will not be specific to COVID-19 because we have not specified it here. We had just said coronavirus spike protein, so it will give us different spike protein for different coronavirus that exist. So if I, for downloading, just for showing you how to download a sequence, I'll just select bovine coronavirus, which is the first hit that has been given. Okay, so this is the page that you get. Here you will get all the information required for the RNA. Now suppose now I'm talking about RNA or DNA or the protein for that matter. Whatever you need, that is what was the source, what was the organism, what are the coding regions for different genes. So now because this is specific for the spike protein, if we select the whole sequence is selected, if you have the whole genome instead, then you will get the only, when you select spike protein, only portion of the sequence will be selected. Now downloading the file, usually FASTA format is used. So you click on FASTA over here to go to the FASTA format of the sequence. So this is the FASTA format. As mentioned in the tutorial video, the FASTA format has this definition line. In this definition line, this bracket is very important. If in case you are copy pasting the sequence, and while doing that, if you delete somehow, this bracket gets deleted, your sequence will be not identified as a FASTA format. And that's why some of the bioinformatic softwares may give this as an error. Now how to download a file. So click on send to, then you want, of course, the file destination. So it has to be in a file in your computer. So click on file and click on FASTA format. That is the format we want, and create file. So I've already downloaded a file earlier, and this is what it will look like. So you can open it. So while saving it, either if you save it as FASTA, then it will be saved as .fasta. But if you want to save it as a TXT file, which can be read with a normal Word document also, you can go to all files, and while naming, just put at the end .txt and save it. So once you save it, your file will get saved and you can open it later on. So this is how the save format looks like. It's either this, or you can just open the notepad, copy paste the sequence into your computer and save the file. So these are the two types, two ways that you can save your sequence. Any doubt if someone has, or am I going too fast? You can.
Akanksha, you can continue, and I think other team members, Arup and Manisha, please reply in chat box.
So if you want to do a multiple alignment, then either you have to be ready with your sequences that you want to align, because you have to do a literature review before going for any experiment. So if in case you're, so this is the spike protein for coronavirus. So if I'm studying about coronavirus and I want to know how the spike protein has evolved over the different mutations or different animals, then what I will do is, before multiple sequence alignment, I'll make a file wherein I will paste one after the other all sequence. So suppose this was SARS-CoV-2 sequence. The next sequence I will copy paste is of bovine coronavirus. Then if I want to talk about MERS virus, then Middle East respiratory syndrome related coronavirus, like that I will copy paste and make a file and keep it ready. So now that we are ready with the files, we can now go to the BLAST tool. So BLAST tool is also, you can search it on Google, and it is also maintained by NCBI. So it will help us find the regions of similarity between the sequence, and will also tell you how statistically significant it is. It is not. It will not give you back results which are similar just by chance. So what you do here, enter query sequence. So this query sequence can be the sequence that you have just downloaded, or either the sequence that you want to study. So I'll go back to my, I'll get SARS or beta actin, which is a very common protein in humans, and also is used as a control protein for many studies. I'll select this protein and paste the sequence. This is one way, or the other way is I choose file and upload the file that I want to BLAST. The next set of parameters is what database I want to choose. So for now, I just want to look at the nucleotide collection. Next, if I want to exclude any organism, BLAST let me do that. If I want to exclude a certain type of sequences, and what kind of, strictness should the BLAST follow, whether it should only look for highly similar sequence or somewhat similar sequences are also okay. That is for the program selection. Now let's keep all the settings default and run the BLAST. Usually this does take time, and it's taking time. So now this is how the page will look while it is running. And this is the result page. So here, section is just the information that you have given as an input. The job title, the length of the sequence, what was the DNA molecule type, etc. Then you can filter your results. So either it's you can include the result that you want, can include only names of some animals, or you can exclude some animals. Then it will also let you filter according to the percent identity, the e-value, and the query coverage. So now if you see, here is the table results that you get through BLAST. So as you can see, this is the score. This is given based on the matrices that goes behind the BLAST tool. The e-value, basically what at this level, what you need to understand is the lower or the closer the e-value is to 0, the significant your results are or the similar your sequences are. Then you have the query coverage. Now, query coverage means if you have a protein or a sequence, and then BLAST is aligning another sequence with it, how much of the sequence is like, how much of the query that you have entered is covered by this sequence. So for humans, of course, it should be, it will be 100. But if you go to other animals, due to the changes in the sequences, you can see that the query coverage is not 100% for all. Then there is percent identity. So percent identity again is like when you align two sequences, how much of identical they are to each other. So this is what you get for all the results. Now let's go to the graphical view or graphical summary of the results, wherein this is the query. Now this query means the sequence that you have submitted. Next comes the different, so in the tabulated results, you have seen that these are the different organisms that were tabulated over there. Now red means a score of over 200. Here these lines which are gray are actually gaps. Pink is a score below 80 to 200. And if you can see here, there are some black lines, and these can be the places where the alignment scores are lower than 40. So that way, you can say how the distribution of top 500 BLAST hits on 100 subject sequences. So these are 100 subject sequences that the BLAST has analyzed. Then you can go for alignment. So here, what it does is, it will give you your sequence. So this is query. That is the sequence that you have given as an input. And this is the other sequence, that is Homo sapiens beta-actin ACTBT sequence that has been aligned with your query. So this alignment will be given for all the sequences that were there in the table. Next, we can go to taxonomy. So taxonomy is basically, it will tell you how these, so taxonomy and lineage will group them on a tree. Okay. So this will tell you how far away from each other they are in taxonomy. So this is the basic BLAST result that you will need to know at this level. And when researchers do perform BLAST, there are several other filters and other criteria that they see, which will like specifically to their experiment. Now, BLAST help videos are also there. These are again maintained by the NCBI, which can help you if query, and they also tell you how to read this report if you want to go back to the results and etc. So that's it for BLAST. Now next is multiple alignment, and what we use is Cluster software. So Cluster Omega, which is maintained by EMBL EBI. So this is the web page for Cluster Omega. First you enter what kind of sequence are you adding. If you're adding a protein or if you're adding nucleotide sequence like RNA, DNA, it will let you choose. For now, I will show you how to do it with protein. Then they will ask for sequences in any supported format. So this can be either you copy paste the sequence one by one here in this text area, or you can choose a file. So what I'll do is I have already created a file for this session, and I'll be I'll just select the file. Then these parameters are the output format that you want. So I'll keep them as default. And there are other options also, but at this stage it is not needed. And then I'll submit it. So this is how the result page look like. First, it will give you an alignment, then the result summary, which is the input sequence, the tool output, etc., and the percent identity matrix. Then it will give you a guide tree, which is like a phylogenetic tree. This is the cladogram, and if you can also choose for a real phylogenetic tree, I'll explain it to you later. Then again, a phylogenetic tree, and it also has its own result viewer, which is the JAL view, but not required at this stage. So if you go back to the alignment, for proteins, you can also see colors. So these colors are basically dependent upon the different properties of the protein, that is acidic will be red and etc. Now, what you need to understand is when you are going through, so now what samples I have taken for the sequences that I have taken for multiple alignment are avian coronavirus, bovine coronavirus, the MERS coronavirus spike protein, the pangolin coronavirus, SARS-CoV-2, that is the COVID-19 causing virus, RATG13, it's a variant of bat coronavirus, SARS-CoV, and again a variant of bat coronavirus. Now, if you see this, if I zoom onto it, at this place, all the sequences have P as a protein. That means this place is very highly conserved over the course of evolution. Now, if you are to design primers, then having such sequences which are highly conserved won't work if you want very specific primers. But these regions will help you understand how the infectivity or the if you are studying the role of mutations or role of evolution in the infectivity of virus, then these conserved sequences will be really very helpful. And then we go to the phylogenetic tree, which is another thing that will help you understand your results. So a cladogram is basically a phylogenetic tree, but it will not show you the difference in the distances between them. It will normalize all the distances and show you. So all the branches will be of same length. But if you go click the real cladogram, that will show you the difference in the distances. So as you can see, SARS-CoV and SARS-CoV-2 and the bat variant are very and the pangolin one are placed close to each other. But if you see avian virus, it is not placed close to the SARS-CoV-2 virus, and the bovine and the MERS-CoV-2 virus are placed very close to each other. And this has also been seen in the literature or published. This is already given facts that MERS and bovine are very placed very close to each other, and they have more identical sequence similarity than the SARS-CoV-2. So this is how a cladogram will help you mention. So if suppose you have a protein or a sequence that you have just discovered, then if let's say the protein is placed somewhere here, so you will know that, okay, the protein is quite similar to SARS virus, but it has less sequence similarity with that of SARS-CoV-2. And this is how you can interpret the results. Now, after you know that how similar your sequences are to other sequences or other strains of the viruses, now you can finally go and decide, okay, what part, what sequences can be good for specific primers. So one primer designing tool, again maintained by NCBI, is the primer blast, wherein so first earlier what used to happen is we used to use a tool called as primer 3, and then blast the sequence which we have, like the primer sequences that we get using primer 3, to search if the primers are specific or not. But now it has been made easy for us, where primer blast, it will also search for your sequences and give you the results and the end results. So I'll take SARS-CoV-2 gene and select. So all these software or online tools will give you two options, either to copy paste your sequences or upload the file. Again, if you want, suppose let's say if you're looking out, there's a human protein I'm working with, and I know that I have to start with nucleotide 100 for the forward primer, and end with 125 and everything. Like, you know the range that you want, you know the gene product that you want from the sequence. At that time, you can specify what is the range, like this range. But now we don't want that. Okay, if you have a reverse primer or a forward primer, but you want another the other set to be completed, then you can give out what is the size. So the product size minimum 70, and I'll say keep it 200, and primer pairs to return. So this will tell, this is actually the tool is asking us how many primer pairs it should return. So should we say 10 or 20, depends upon depends upon your experiment. Then the primer melting temperature. So this is okay, let's keep it default. If there are any exons. So what happens is, if you have a exon in the sequence, then those won't be like, this is a little, okay. If you have a mRNA sequence and then there is a exon span or exon which divides the sequence, then you don't want that to happen. Then you can give out like specify whatever you want, your intron inclusion. Then, okay, these are all very specific things. We'll keep it as default, not required at your level, and get the primers. Okay, this is taking time.
Akanksha, this is the last thing which you are showing probably now, right?
Yes, yes, sir. It's taking time.
That's right. So I think since the all the sequence detail, everything is given to the participants. We will also upload this video to everybody. They can do that. But I think some of these software tool can be taken like what you are showing. Design the things, looking at many considerations, right, including the TM value, GC value, length. Many stuff can be taken. And now I think you can explain further that lot of possibilities are here to choose.
So yeah, this is what the primer blast results look like, and they give you a graphical view of primer pairs. So when this is the template that you have provided, and these are the primers and where they go and bind. Okay, so this is like just the graphical overview. Now comes the important part. So first primer pair, let's discuss in detail. This is the forward primer, reverse primer, of course. Then the length of the primer. So every primer is, both the primers are 20 base pairs long. What is the starting base pair and the stop base pair of the primer? And the melting temperature. So now what is melting temperature? Melting temperature is the temperature at which the two strands of the DNA like separate. Okay, and usually this is the temperature. Annealing temperature is 5 degrees above or below the melting temperature. So now, depending upon the experiment that you are setting up, you will select which primers will be good for your melting temperature. Melting temperature, according to the melting temperature, which primer will be good. Then you have the GC content. GC content is basically the GTP, the guanidine nucleotide and the cytosine nucleotide content. So mostly it has to be at 50. But 55 is also okay, but don't like it has a difference of 5% is okay, but not more than 5%. Then self-complementarity. Your self-complementarity means the possibility of forming the hairpin loop structure. Now it should be low, and this is quite high. If you go here, this is a good result. Then self 3' complementarity. So self 3' complementarity is the possibility of forming primer dimers and then polymerizing itself. So it should be also low as possible, but 2 is fine. Then you of course need to know what is the product length. So depending upon all these primers, you will select which one are best suited for the experiment that you're designing, and then take it ahead with the company or whatever vendor you are asking to produce primers for. That's it. So this is what the basics will cover, and I am done. Thank you.
All right, Akanksha, thank you very much. I think you have made very informative session. This could be slightly difficult for people who are just entering in to do the bioinformatics, but I'm sure for even people at the advanced stage, they would find it useful. So all the participants, I think now get ready for starting your quiz today. Today we are, since we realize conducting 3 quizzes in the online mode of Kahoot is taking a lot of time. So we are going to give you Google form, right? And that will happen simultaneously. So you can only choose one, which belongs to your category. So we will be now going to share you the Google form. You will have 20 minutes to do that. Today's session recording, I am going to upload that in the same playlist of the workshop or the summer camp. And also I'm going to put the Google Drive link over there. Please do watch it again in case you are not able to follow the pace of it. And then try to go through even the previous tutorial video which we have uploaded. Again, I'm sure you can understand that a lot of exciting and interesting things can be done at your home and sitting at your place, which if you don't do it yourself, you can't understand what we are talking, right? So you have to make some effort. And if you can watch those videos and today's live session, do some of these activity yourself, you'll be very much well prepared for the actual summer camp session when we'll be giving you some of the live assignment and questions, and then you can participate actively over there. That's all for today, everyone. We will announce the name of the winners by email, and also we will announce that tomorrow in the session. So tomorrow again at 3 p.m. we have next tutorial session, which will be more about looking at the drug docking and how to do some of the the docking experiment for protein drug interaction or protein interaction, those kind of activity, which is very interesting happening right now. Yeah, also in the course of drug repurposing aspect of the COVID-19. So we will take some of those activities. What we request all of you, please put your question in the chat box because we can't take it now in person. And we are going to start the quiz shortly. Okay. Thank you, everybody.